In this short video, I would like to walk you through the metabolism of aromatic amino acids, phenylalanine and tyrosine. So, phenylalanine is an essential amino acid that has to be supplied in the diet. As the name says, it has got a phenyl group which is attached to the alanine part. And this phenylalanine is acted upon by an enzyme called as phenylalanine hydroxylase. And for this enzyme to add the hydroxyl group, it requires a source of hydrogen, that is NADH, and it also requires a source of oxygen, that is molecular oxygen. And this reaction takes place in the presence of tetrahydrobiopterin, which has got a structure similar to that of the vitamin folic acid, in that both tetrahydrobiopterin and folic acid have a pteridine ring in their structures, and however this tetrahydrobiopterin is not a vitamin, and this tetrahydrobiopterin is synthesized from GTP. And what happens to the product? The product is nothing but tyrosine, which is also called as para-hydroxyphenylalanine. As a result of this reaction, a hydroxyl group is added to the para position of phenylalanine to give a product called as para-hydroxyphenylalanine, also called as tyrosine. And this tyrosine is acted upon by an enzyme called as tyrosine aminotransferase. This is similar to the transamination reactions that take place in the other parts of the body. The amino group of tyrosine or para-hydroxyphenylalanine is transferred to the keto acid called as alpha ketoglutarate to give the amino acid called as glutamate, and in the process tyrosine is being converted into para-hydroxyphenyl pyruvate. And this para-hydroxyphenyl pyruvate is acted upon by an enzyme called as para-hydroxyphenyl pyruvate dioxygenase, and for this enzyme to function it again requires molecular oxygen, and we get a closed ring structure called as homogenticate or homogentic acid. Now this homogentic acid is acted upon by a homogenticate 1,2 dioxygenase, which again requires molecular oxygen, and the product that we get is mallyl acetoacetate. The ring is broken. The ring of homogentic acid is broken, and as the ring opens we get a structure called as mallyl acetoacetate, which has got 8 carbon atoms. The mallyl group has 4 carbons and acetoacetate group has 4 carbons again. And this mallyl acetoacetate is being acted upon by an isomerase, which gives a product called as fumaryl acetoacetate. Both the structures are cis trans isomers. And again this fumaryl acetoacetate is an 8 carbon compound which is being acted upon by an enzyme called as fumaryl acetoacetate, also called as fumaryl acetoacetate hydrolase. So this reaction uses water to break the bond between fumarate and acetoacetate. As a result of this reaction we get two products, a 4 carbon compound called as fumarate, which is glucogenic, and another 4 carbon compound called as acetoacetate, which is ketogenic. And coming to the inborn errors of phenylalanine and tyrosine metabolism, the first and most important disorder is PKU or phenylketonuria, where the enzyme phenylalanine hydroxylase is deficient. The second disorder being tyrosinemia. There are 3 types of tyrosinemia. Tyrosinemia type 1, where fumaryl acetoacetate hydrolase is deficient. Tyrosinemia type 2, where tyrosine aminotransferase is deficient, and tyrosinemia type 3, where para-hydroxyphenyl pyruvate dioxygenase is deficient. The next and the most important disorder is alkaptonuria, where there is a deficiency of an enzyme called as homogenticate 1,2 dioxygenase. Okay, what we see here is the synthesis of physiologically important products from the amino acid called as tyrosine. And in this pathway tyrosine is being acted upon by an enzyme called as tyrosine hydroxylase, which adds the hydroxyl group to tyrosine to give the product called as dihydroxyphenylalanine or dioxophenylalanine, also called as DOPA. And this hydroxylase, like phenylalanine hydroxylase or tryptophan hydroxylase, requires molecular oxygen and tetrahydrobiopterin to function. And what happens to this DOPA or dioxophenylalanine or dihydroxyphenylalanine is that it is being acted upon by an enzyme called as DOPA decarboxylase. In this reaction carbon dioxide is removed and we get the corresponding amine called as dopamine, and this dopamine is being acted upon by a dopamine beta hydroxylase, molecular oxygen is being used in the process, and the product that we get is norepinephrine. The term nor here means that there is no methyl group attached to this structure. And norepinephrine can also be called as phenylethanol-amine. Now a methyl group is transferred to this structure called as norepinephrine or phenylethanol-amine, and the donor of this methyl group is S-adenosyl methionine, and in the process S-adenosyl methionine gets converted to S-adenosyl homocysteine after it donates its methyl group to norepinephrine. And from norepinephrine we get the biologically important compound called as epinephrine, and this is the methylated product of norepinephrine, and it is the hormone of the adrenal medulla. And dopamine, epinephrine and norepinephrine are the three catecholamines that are synthesized from the amino acid called as tyrosine.